Welcome back to this lecture series on the neurophysiology for the human systems, human nervous system. Again, we are considering neurons, the cellular and network properties. This is the second in the triplet set, if you will. You will find that the 8A ended up being split into 3, even though during the recording I said it would be 2. This one, I assure you, will only be a single one. There are fewer slides. And what we're going to consider as the chemical signaling in this discussion, even though interneuronal junctions can have electrical signals passing straight through with gap junctions, we're really interested in a chemical synapse for this discussion of the cell-to-cell communication within the nervous system. Obviously in the context now of neurophysiology. But we had some of this in consideration with the topics of neuroendocrinology, and we'll just have a few distinctions as we proceed here. So the chemical synapses use neurotransmitters, and of course the electrical synapses pass electrical signals. So we recall the integration at the dendrite and the soma, converging, on the axon hillock, the spike zone for these multipolar neurons, and then triggering action potentials that are conducted to the terminal. And now we're at the terminal to consider some of the characteristic features, anatomical and mechanistic, that we find there for this digital, remember the action potentials are digital, to analog signal the full range of concentrations of the neurotransmitters within the synapse. And then for any signal, it must be initiated or originate, be present to have an effect, and then disappear, otherwise it's no good, right? So if you're going to send a message with a light, as you probably have seen in some of the old military movies where in the Navy they used the light to flash it, and use Morse code, I guess is what they had used for their coding signal. If you just leave it open and on all the time, there's no signal there, right? So there has to be a start and a disappearance. So we're going to consider that feature of the mechanism for intercellular or cell-to-cell signaling in the nervous system. So again, we recall the axon terminal is called the presynaptic terminal because it is on the axonal side of the synapse. The interneuronal junction is what we are really considering here, although there are others. And the synaptic cleft is the space immediately between the axon terminal and the postsynaptic membrane that might be on a dendrite or the cell body, or even a portion of the postsynaptic neuron's axon, an axo-axonic type of a synapse. So the action potential will be carried along the membrane, and we already talked about the presence of the calcium, voltage-gated calcium channels being associated with the exocytosis, and a certain number of vesicles. It's called quantal release. For every action potential reaching the terminal, a certain number of calcium channels will open. A certain amount of calcium, number of calcium ions will enter locally here at the presynaptic membrane, and a certain number, a quanta of vesicles will fuse and dump their contents into the extracellular space that is confined here. Remember, wherever you don't have neuron contacting neuron, there's astrocytes. All around here are the astrocytes. Where if we go above here, where the if this is the CNS, where this portion, myelinated portion of the oligo on the axon ends, then all this surface area is covered with astrocyte processes. So they leave it white and open, making you think that there's just this gigantic interstitial space, but it isn't. It's all occupied by astrocyte cellular processes, so there's just a very, very thin film of extracellular fluid surrounding all of these structures. So we'll talk about the escape from the synapse of these transmitters. And obviously in a situation like this, they're just going to wash away into this big open space. Well, that isn't what really happens. The cells are packed together very tightly. So let's move on to the next slide. We see here the cell-to-cell events at the synapse and exocytosis are being shown. And there's a series of numbered events, 1 to 4, and we're going to go through each of these as we proceed. So let's just summarize what we're seeing here. Again, we're seeing the presynaptic terminal with the presynaptic membrane and vesicles at various different stages in the process of exocytosis, and there is transmitter and presynaptic ion channels and postsynaptic proteins that will be there for the receptors to mediate the cellular response. And we have the synaptic cleft. So there's a certain volume of interstitial fluid in the synaptic cleft, and it's within that volume that we contribute the transmitters and remove them, and so it's the average over time, the concentration of transmitter in the synapse that determines the likelihood that any one of them is going to bump into and hit the binding site on the postsynaptic receptor and thereby have an effect. And remember, we talked about over a period of time what is the likelihood, the probability that you're going to see this receptor in the ligated form such that it would be in the active state more often. Okay, let's begin then. So the action potential depolarizes the axon terminal. So we see that number 1 coming down here, and the presynaptic membrane is red, and action potential has reached it. That is going to cause a depolarization of the membrane. So the voltage sensitive portion of the voltage gated calcium channels is going to undergo a conformational change, and the gate is opened, right? And the calcium is higher extracellularly than intracellularly. We covered that early in earlier talks. You cannot have calcium floating around inside the cell at any area. It is damaging. So it will enter and produce a signal, bind to a series of proteins, so it's no longer free cytosolic calcium, and then bring about the events that we're going to follow here. So again, we said if we could have antibodies that would fluoresce and stick to these cells to show us where proteins are in the axon membrane, we would see a high density of voltage-gated sodium and potassium channels and sodium-potassium ATPase all the way out to the presynaptic membrane as well. And then the voltage-gated calcium channels would really light up. There's not just 2 of them, obviously. They are scattered all around, but at a higher density here than pretty much any place else on the, especially the axon, because they're going to be coupled to the intracellular vesicular release mechanisms. And we see that indeed is the case. The calcium will trigger exocytosis of the synaptic vesicle contents through a series of proteins, and then those proteins interact with the cytoskeleton to bring the vesicles close to dock. You see the docking protein is shown here, and then ultimately there is an opening. So there's discussion in the textbook about whether this membrane is inserted, and then at another location, as we talked about in the first lecture of this series, where you have endocytosis to recycle the membrane and generate new vesicles from old membrane, but anyway, new vesicles that will have to be reloaded with transmitter and be available then for release. The textbook also discusses the pool of vesicles that are here. There are some that are very close to the presynaptic grid, as it is called, that are readily available. They're closest, so they're going to be the first to come in and dock and undergo the exocytotic event. There's another current hypothesis or theoretical model that has developed that purports that there is an attachment and a pore that is formed. That's the kiss, and then this will simply close off again, and then the empty vesicle will release and then go through a recharging, recycling phase to become available. And they kind of make it look like it floats around like this, but remember, these are just coming on and off and on and off locally all along this grid. These reduced illustrations should not mislead us. If you've never looked at an electron microscopic image of an actual synaptic terminal, you'll see one on either a quiz or an exam, so I encourage you to seek out that type of an image and become familiar with it. So now we have again, there's nothing inside the vesicle just like squeezing it and squirting it out like a tube of toothpaste. It just is a concentration gradient. So you're going to have the high concentration inside the vesicle and the lower concentration in the synapse, and this will diffuse out into the synapse. Now, if this really is inserted into the membrane, obviously when this flattens out, it is ejecting. There's a physical ejection or displacement into the synapse. So whichever way you want to think about it, we have effectively contributed more transmitter to the fluid, the extracellular interstitial fluid within the synapse, and that should have increased the concentration there, meaning that there are more molecules of the neurotransmitter, the chemical signal, available to randomly bounce around and bump into anything. That's how they're going to diffuse around, and they might, diffuse, try to get out of the synapse as well, and we'll see that in a moment. And that spread outside of the synapse is important too. Early in the theoretical modeling of physiology, that was not considered to be of any importance. It was basically taught that they did not escape from the synapse. Now we know they do, and that there are extrasynaptic, non-synaptic receptors that can bind those and are basically being stimulated with a signal saying the synaptic concentration is so high that it's diffusing out here and binding to you. So the term for that is volume transmission. It's not so critical for you at the moment, but I do like to build on that bigger picture. So think of the concentration, early discussions, review lectures on diffusion. You put a drop of red dye in here, they randomly bounce around and begin to move farther away from each other, and they randomly bump into the receptor in all different parts of it. Those that happen to hit the binding site will bind for a period of time and unbind, and then another or it will be right back on it. And as you increase the concentration, this is occupied more frequently. As you decrease the concentration, it is occupied less frequently. And that's the way the system works. And the cell response is cumulative based upon that frequency of occupation and activation of the receptor. So it isn't the simple thing of the neurotransmitter binding and initiates a response in the postsynaptic cell. It is that frequency event. So when a drug is administered that's going to act at this receptor, let's say an antagonist, to prevent the agonist, the endogenous agonist, from having its action, say it's someone where you need to slow their heart rate for whatever reason, then you want to give a dose that ultimately, when it diffuses through the whole body and finds its way into synapses, will produce a high enough concentration in here so that its relationship to this, as it's randomly bouncing around along with these molecules, if it binds here, hopefully it's developed so it has a higher affinity. It likes to stick to the protein more than the endogenous ligand, and it's going to hang there a little longer before it lets go, and another one of its molecules, or it, will bind again before the endogenous ligand will. But at any one point in time, this may be unligated or ligated, either with the endogenous ligand or the drug that was administered. And the dose of the drug should be sufficient, so that it occupies this binding site on the receptor more frequently or at a frequency to alleviate the symptoms that is the objective of your drug treatment, correct? So drugs of abuse that act here mess this system up. They confuse it a lot, and that's a big part of the challenges for rehab and for the understanding of the biology of drugs of abuse. So we are at number 5 for the cell response. And that response has to be terminated too, right? If you're talking about a drug binding to receptors on your SA node that are making heart rate increase, obviously you want that to be terminated at the end of the startle response, right? So you're sitting studying and somebody comes in and says, good morning, and you jump, right? You get a bunch of adrenaline going to the heart, epinephrine, sorry, can't overcome those old terms, and heart rate goes up. Well, you want it to be terminated, so we're proceeding to ask that question. How do we terminate the signal? Otherwise you're going to drop dead, right? Every time somebody startles you a little bit. So this type of cell-to-cell interneuronal communication is referred to as neurocrine. So it's not endocrine, but it is between cells, and it's using the extracellular fluid for the communication. So that's that crine part with the body fluids. Early on, these were things that humans discovered. When we, you cut somebody and they bleed. There was a lot of war going on, a lot of events, trauma, and so forth. Humans being eaten by animals, I guess. So we knew there was something liquid that came out of our bodies if we were not the ones that got eaten, right? So that's kind of the origin of this crine of like things are floating around in our fluids and having effects. So this is the neurocrine terminology that has emerged to describe this mechanism of interneuronal communication. So there are 7 classes of these neurocrine or neurotransmitters. There's the acetylcholine, kind of unique in and of itself, modified amines, and amino acids themselves, purines, gases, peptides, and lipids. So there's just these. There may be others, but these are the essential ones that we know the best or know at present. So there is a table in the version of the textbook, the edition of the textbook that I'm using presently, and I presume that any time in the future there would be some similar type of table presented in the textbook. So as we proceed through these slide presentations, I was wishing the table would be there, but I'll pull a few points from that table as we proceed, and quite obviously the table would be a source of questions for quizzes or exams, so you need to familiarize yourself with the details provided in those tables. So the cell-to-cell communication, we're now thinking of the synthesis and recycling of acetylcholine at the receptor, and this discussion introduces the first mechanism by which the signal is terminated, and we see that if we go quickly directly to the postsynaptic membrane, an enzyme is expressed on the surface of the postsynaptic membrane called acetylcholine esterase. So it obviously breaks the ester bond between the acetyl group and the choline group, acetylcholine. So we'll talk about the synthesis now, since we want to put the molecule together after we realize that the basic components are some type of acetate or acetyl group and the choline group. So we know that choline is common. Phosphatidylcholine is one of the phospholipid molecules found within the plasma membrane. So choline is common. in our tissues and in the mitochondria. We know we can have the production of the acetyl-CoA from the pyruvate, from the glycolytic pathway. The pyruvate moves into the mitochondria, as you recall, and acetyl-CoA is produced, and this is providing some acetyl-CoA in the cytoplasm as well, not directly, it's an indirect method, but anyway, we know that acetyl-CoA can be produced within the cell. Usually it's used for energy. In this case, it is used in the production of a neurotransmitter. And this enzyme, they don't have it labeled, it's number 1. It's made from choline and acetyl-CoA, so choline and acetyl-CoA are the substrates. And this enzyme is choline acetyl transferase. So it transfers an acetyl group from acetyl-CoA to the choline, and then you have acetylcholine. Then that is transported by a vesicular transport protein, VCAT, I think it's called, vesicular choline transport, VCAT, right? That would make sense. And that loads the vesicle. I mean, they're showing one molecule of acetylcholine in there. Obviously, it's more than that. But anyway, it helps us to picture what is going on. And under normal metabolic conditions, the same amount of acetylcholine would presumably be loaded into each vesicle. So there's all the saturability features of that, and you'd presume to have more than just one vesicle in a synaptic terminal. There'd be a population that would be considered to be homeostatically regulated as a stable pool of the readily releasable pool and some backup pool that's a little bit farther back here that can be pulled in if you are, like when you're exercising, you know that acetylcholine is at the neuromuscular junction. So when you really exercise extensively, you can deplete the vesicles in your acetylcholine neurons. If you can overcome the homeostatic mechanisms that sustain your readily releasable pool, your backup pool, and the recycling, then you deplete them and you reach a point of exhaustion. Right? So it's possible to do that. Also, the muscle and the neuron is being very active, so they're going to want the acetyl-CoA to produce ATP, so there's competition there. And just before proceeding with this, one clinical note is that there's some hypotheses about this interplay between acetyl-CoA and acetylcholine synthesis and the need for acetylcholine to produce energy and how efficient mitochondria might be in aged or neuropathological conditions, in particular with the condition of Alzheimer's disease. As you might know, or will know now, acetylcholine is one of the first neurotransmitters to decrease significantly in Alzheimer's disease and is directly correlated with the initial losses of cognition, cognitive function, and even the more advanced progressive symptomology of more complex cognitive deficits, not being able to form new memories and things like that. And as you also may know, one of the only approved treatments to delay the symptoms of Alzheimer's disease is the regular use of acetylcholinesterase inhibitors. So whatever is causing lower amounts of acetylcholine to be present, if you decrease the rate at which it's removed from the synapse, then the synaptic concentration should be sustained at a more normal or closer to normal level than would be otherwise in the diseased or pathological condition. So any other drug used in Alzheimer's disease typically is just to suppress the person. They're mostly depressants that are given, benzodiazepines and things like that, to relax the person. I don't know if you've ever worked or been around Alzheimer's patients, but if you have, you'll understand that. part of the cognitive deficit is not being able to make sense of your environment and put it into present context, and therefore to suppress aggressive and defensive behaviors, so or responses. So the response to the environment that can't be understood and put into the appropriate context is inappropriate for the environment. So clinical management becomes a real challenge. So even though you might be sustaining acetylcholine levels, the neurodegenerative diseases progress to the point where the neurons just aren't there. The synapses aren't there to keep the acetylcholine in, and so you have to use some other type of drug management system. And it is justified. You may be thinking that should never be done, but if you are ever in a situation in that type of a clinical setting, or have a family member in the distress of just a normal environment, you will understand that justification. So obviously then the exocytotic release, as we had discussed, and then producing a synaptic concentration, and then all the likelihood, probability things that we had talked about for binding to the postsynaptic receptor. So to go to the table quickly then, the acetylcholine receptor, I think it might be discussed later, but I'm just going to bring it up at this point. There are two types, nicotinic and muscarinic. The nicotinic is an ion channel receptor that allows sodium and potassium through. That would be what you'd find at the neuromuscular junction for sure, and not so commonly within the central nervous system, but definitely the nicotinic, as we will study with the autonomic nervous system, is present between the presynaptic and postsynaptic neurons. And they are presynaptic and postsynaptic, but they're preganglionic and postganglionic neurons, not to confuse the terminology quite yet. And obviously nicotine is an agonist. Curare, alpha-bungarotoxin are common antagonists. So curare, is one of the toxins that the natives in the jungle put on the tip of their blow darts to immobilize their prey so that they can capture them for their food, right? So the muscarinic receptor is what we might imagine this receptor would be, and this is one reason for bringing it up at this time. That doesn't look like an ion channel, so that very likely would be a muscarinic, which is a G protein coupled receptor, even though they're not showing it. This is more typical appearance of a GPCR, G protein coupled receptor. The GPCR acronym is what's used in the table. So we'd find that in smooth and cardiac muscle, so the targets of the postganglionic parasympathetic neurons, and the endocrine and exocrine glands, again, the postganglionic autonomic parasympathetic target cells. And it's the muscarinics that we are more likely to find in the central nervous system. Excuse me. Even though I should go back and clarify, there are some nicotinic receptors distributed throughout the central nervous system too. And hopefully you corrected me on that back there, at least saying, well, how do people get so addicted to nicotine if there's no nicotine receptors in the brain, right? So obviously there's some nicotine receptors in the brain, and they are involved in a lot of circuits, some of which are reward and reinforcement, and that's how we get addicted to substances that act within the nervous system like that. So in the nervous system, individuals in the CNS, individuals that use nicotine, wherever so I think the nicotinic receptor tends to be presynaptic and have other kinds of functions, but regardless, wherever it is, the nicotine that is consumed by nicotine users enters the synapse from the interstitial fluid, from the plasma, however it gets to the plasma, Nicorette gum or inhaling cigarette smoke, and produces or accumulates here in the synapse and produces a certain concentration. And then it begins to, I don't want to say competitively, but I guess it is. It's like a competitive agonist. If it's at a higher concentration than the acetylcholine, then this receptor is going to be occupied by nicotine more frequently than by the endogenous ligand, the acetylcholine. So let's move on from the acetylcholine. Hopefully that gives you some insight on how you can just talk about one transmitter from an illustration like that and obviously carry it to many different levels. Well, let's now think of the amines. So they are derived from single amino acids. One example is tyrosine. So from tyrosine, the catecholamines are produced: dopamine, norepinephrine, and epinephrine. So from tyrosine, there's a whole series of enzymatic conversions to produce dopamine, and then dopamine is converted to norepinephrine. It's an enzyme called dopamine beta hydroxylase to produce the norepinephrine. And then the norepinephrine is converted to epinephrine by the enzyme phenylethynolamine N-methyltransferase. I had discussed that in the endocrinology lecture because we saw these two chemical signal substances related to the adrenal medulla. Others are serotonin from tryptophan. So you might have heard of people that take tryptophan supplements or 5-HTP supplements. 5-hydroxytryptamine is tryptophan. Right? No, wait. 5-hydroxytryptamine is serotonin. 5-hydroxytryptophan is the intermediate between tryptophan and 5-hydroxytryptamine, serotonin. So some people take, a lot of people used to take tryptophan, an amino acid, to impact their serotonin levels. Whether or not that's true, there's some evidence for it. But not everyone is completely convinced by any of the evidence. But anyway, a lot of people were taking it, thinking it might help them sleep at night, a somnolent, similar to the melatonin that we talked about before. And then there was a problem with it. It was being produced in bacteria, and some toxin got in or something like that. So tryptophan is no longer it may be coming out again now, but anyway, it came off the shelves for a long time. So people started marketing the intermediate, the 5-hydroxytryptophan, 5-HTP. So you can probably see this on your markets. I see it at the HEB pharmacy when I'm looking for vitamin A and vitamin C and stuff. The 5-HTP is there alongside the melatonin and things like that. And so that is taken thinking that it is going to contribute to the production of serotonin. Now, collectively, although it's not shown on this slide, those amino acid derived transmitters from tyrosine and tryptophan are categorized into another subset that's collective for all of them, called the monoamines. So you may hear of monoamine inhibitors or just monoamines in general. And what that's referring to are this set of catecholamines and the serotonin transmitters that are generated from tyrosine and tryptophan. And histamine is made from histidine. So histamine is a major CNS neurotransmitter. If you don't believe that, take some Benadryl or any of the antihistamines that readily cross the blood-brain barrier and see what happens. So if you take loratadine, is that the name for it, right? Which I take, and I can't remember I remember loratadine, but not the common name for it. Anyway, it's obviously derived from this term, the histidine. That is designed so that it does not cross the blood-brain barrier at the dose of 10 milligrams. is an effective peripheral histamine receptor blocker and does not have the central effects, so it doesn't have the warnings on it that you should not operate any machinery or things like that. So I like to take this information and put it into place, and hopefully it helps you to think about it. So let's think about now the category of the amino acids that are acting directly as transmitters. So glutamate is a major one, and we'll see more and more of it as we proceed. It is the primary excitatory neurotransmitter, the fast excitatory neurotransmitter that we find throughout the CNS. And aspartate is another that you don't always hear so much about, but it also is a widespread excitatory transmitter of the brain. And the primary inhibitory neurotransmitter found throughout the nervous system is gamma-aminobutyric acid, GABA. And it is derived enzymatically, actually, from glutamate. So I'm not sure why it does not... I guess it's a modified amino acid, modification of glutamate, but it remains as a glutamate, as an amino acid. It isn't converted to an amine, such as with the 5-hydroxytryptamine, which is the serotonin. So this is inhibitory. They are showing in this slide just in the brain, but even in the peripheral nervous system, we would find it. There are inhibitory neurons, for example, in the spinal cord that send axons out to inhibit the sensory neurons to attenuate sensory input. And it's found also in a similar fashion for the auditory system. So if the noise is too loud in the background, the GABA will inhibit the primary auditory receptors to kind of blunt it a bit so that you can hopefully resolve the important sounds in the environment, the foreground environment. So it is more widely distributed than just in the brain as indicated here, but it's if you could use an antibody to visualize GABA within the brain, you would see that there's just a tremendous amount of it. Glycine is another amino acid that is found as an inhibitory neurotransmitter, a fast inhibitory neurotransmitter in the spinal cord. Even though there is some GABA in the spinal cord, it's found very heavily in the brain, but glycine is not found acting as an inhibitory transmitter in the brain, but it is selectively found to be and having its own specific receptors that are inhibitory in the spinal cord. Some cases may also be excitatory. So again, it's not so much where you find it and that there is a receptor. Remember, you're not going to have a response to a chemical signal unless there is a receptor. That's the definition of a target cell. Just because a cell expresses a receptor doesn't mean it's going to respond or use that receptor to generate a response mechanism that is exactly the same as in a neighbor cell. So one cell might be excited in the spinal cord by glycine because of the way that it links the binding protein to the response mechanism, and another would be inhibited. So some of the other transmitters then are the purines, such as adenosine monophosphate and adenosine triphosphate. Seems kind of strange that they would be acting as neurotransmitters, but indeed they are sending signals that we would suspect to be relevant to the metabolic state or status of the local area where the activity is occurring. Right? So it's not going to signal the whole brain is too metabolically active, we need to change or something like that. It may just be a local type of an effect. You're concentrating so hard studying for an exam, there's only certain parts of the brain that are active and others that would be inactive. So there's regional variations, as you are hopefully aware, throughout the CNS at different times during different activities. Some of the gases, we already mentioned these, nitrous oxide and carbon monoxide. Peptides such as substance P and the opioid peptides. We had mentioned the opioids versus opiates in previous presentations. Okay, sorry about the dead air time there. I was just taking a look at the table again. So for the GABA, it is. Let me just go back to that. We'll talk about the glutamate receptors for sure, but the GABA is an ion channel receptor, and it is selective for chloride. And so the means by which it is going to be inhibitory is that when it's ligated, it will open the channel, and chloride will move into the cell. There will be an influx of chloride, an inwardly directed negative potential current, and this will make the inside of the membrane more negative than the outside, so it would hyperpolarize. A typical antagonist is one called picrotoxin, Picrotoxin. Alcohol acts through these receptors, and not as an antagonist, but as a potentiator. And the barbiturates also act through this. So remember, we talked about using the barbiturates as relaxants for Alzheimer's patients. That what they'll do is they'll bind to the GABA receptor and potentiate the response when the GABA binds to that receptor also. So the barbiturate would not be acting as an agonist. It would be a potentiator binding to another location on the receptor protein and making it more sensitive to the GABA. So when the GABA binds, you get a more dramatic opening and influx of chloride, an exaggerated response to the same amount of GABA. And then some of the lipids would include the eicosanoids, so we're not going to dwell on that for too long. Now let's think about some of the receptors. So the cholinergic receptors, we mentioned the nicotinic that are on the skeletal muscle and the PNS and the CNS. They are monovalent cation channels, so sodium and potassium. The muscarinic are the ones in the CNS and the PNS, and they're linked to the G proteins. When we talk about the autonomic nervous system or different physiological systems, such as the cardiovascular system, and of course the skeletal muscle system, we'll revisit some of these. So this is the integrated approach that we're using in this class. The adrenergic receptors we've heard about before, alpha and beta, they're linked to G proteins. And as we know, there's an alpha 1 and an alpha 2 and a beta 1 and a beta 2 subtype of each of these classes of adrenergic receptors. Well, now let's think about the cell-to-cell communication for the postsynaptic response in the context of having a fast response and a slow response. And how they're coordinated, and kind of reflect a bit on how they are obviously important. So we're going to go through these step by step in a series of slides. So again, we're thinking of the neurocrine signal between a presynaptic neuron and a postsynaptic neuron. The presynaptic axon terminal is the source, and we are following the fast acting response first over here on the left, so that we see there's a rapid, short-acting, fast synaptic potential. So what this means is that the increased concentration, synaptic concentration of transmitter is transient. So there are terminating mechanisms. We saw one, that there's a degradative enzyme that will inactivate the molecule. One that we haven't talked about yet is, remember, these are just randomly bumping around out here in the synapse. So there are proteins on the presynaptic neuron that are called reuptake transport proteins. So those molecules of transmitter that bind to that will be taken back up into the neuron and either inactivated with inactivating enzymes located inside the terminal, such as monoamine oxidase, right? Would break down the monoamines, the 3 catecholamines, and the serotonin. At the same time, there are VMAT, vesicular monoamine transport proteins, that will be using that same pool that is taken back up into the axoplasm, as it's called. The cytoplasm within the axon is called the axoplasm. So this cytoplasm here will have the molecule that's taken back up, and it's thereby bouncing around, and if it bumps into the degradative enzyme, it's degraded. If it bumps into the VMAT, it's used to recharge vesicles. And so, obviously, those processes, the homeostasis of those, is concentration dependent. The more transport protein there is, the more transmitter will be taken back up. It's a concentration thing, not only the concentration of what's going to be transported, but also the concentration of the transporter. So always keep that in mind, the concentration of the receptors in the postsynaptic membrane and the concentration of the transmitter in the synapse. Both are related to the amount of signal that is going to be generated with any one release event, quantal release based upon a single action potential. Okay, we got it into the synapse. There's more of it there, so you're more likely to have the ligated state of the chemically gated ion channel, the ligand-dependent ionotropic receptor, and it will open or close. It will be opened as it's a ligand-dependent ion channel, and if it's a sodium ion channel, then there will be more sodium that will move in, and this will generate an EPSP, which is an excitatory depolarization. Others, such as the GABA receptor, as we said, which is a chloride-specific ion channel, will allow chloride in, or some will allow potassium out, and this would produce the IPSP. So what I want to do at this time is switch the pointer to a felt tip pen here. And what we're going to do is illustrate what an EPSP, or excitatory postsynaptic potential, would look like, and what an IPSP would look like. So we draw our axis. The x-axis is millivolts, the membrane potential, right? And then the y-axis, the x-axis would be time. And I'm not going to try to spell that if it's going to do those weird things. So we would have resting membrane potential, and IPSP is going to be a positive deflection, right? So that's the EPSP. And then the IPSP would be a hyperpolarizing deflection. So chloride in would make the inside of the membrane more negative, so the deflection would be in the direction of the chloride equilibrium potential, which is minus 63 millivolts. And yes, I did just flip back to that table to look it up. Don't want you to think I remember these right off the top of my head. But we do recall that it is negative, and it's not quite as large as the potassium equilibrium potential. So if potassium were to go out, we would expect, like an equal amount of potassium out. Maybe not shaped like that, but anyway, a greater IPSP than what an equivalent amount of chloride would go out. And why do we say that? Because they are both monovalent ions. Okay, so I just wanted to talk about that at this point because there isn't another option to get very extensively into that discussion in this chapter, and it is important to appreciate that at this time. So these are the fast acting. So we're talking about just milliseconds here, right? And these are these would be the synaptic potentials that would contribute to the local graded potentials, and then the graded potentials would migrate away, as we talked about in the prior lecture. So let's move on to the next slide now. So now we're going to think about a slower synaptic event, the slow synaptic potentials, and the long-term effects. So here's a G protein coupled receptor, maybe a muscarinic. So this could be, if we're thinking in the periphery, a nicotinic receptor that would be the sodium, or in the central nervous system, a GABA receptor that would be chloride dependent, chloride specific. Here now, let's think of a muscarinic nicotinic receptor, a G-coupled protein, G-protein coupled receptor, the GPCR as they use on the table. And here are the G proteins. So as there's a higher concentration, it's more frequently in the ligated form. So you're more frequently going to find the G proteins dissociated from it and migrating toward the second messenger system pathway and converting it from an inactive to an activated second messenger system pathway. And the second messenger is going to alter the open state of ion channels nearby and will either modulate to close these channels or to open and produce the same that we saw before, or modulate the response to the fast one. And in those that might be closed, they would, of course, decrease the current through them, decrease the permeability, decrease the conductance, right? And if you decrease the sodium, then you're going to get a hyperpolarization. If you or a repolarization, typically here at the synapse, you just call it a hyperpolarization. Because it will be hyperpolarized relative to where you were at that moment before this event began. And then less potassium out would be an EPSP, an excitatory depolarization. So what is it that we're thinking about here? This would be sustained for a longer period of time. So let's go to our pointer again, the felt tip pen, and we get our graph, and we want to put a time scale that's a little bit longer here. And what we're going to think about is first this case on the right, let's say, of less potassium out, and it's going to be an excitatory postsynaptic potential. So we are at state 1, whatever it was. Let's say it is resting membrane potential, the minus 70, or it would have to be something a little bit different, where we had some, maybe it's minus 80 because there was some potassium going out. Now we're going to block that, and the membrane is going to move or shift to a more depolarized state for whatever period of time this second messenger system is active, or its activity is sustained, such that it sustains this new state. And at this new membrane potential, closer to threshold, any other stimulus, if we did a stimulus here, is going to be more likely to trigger an action potential that wouldn't look like that. So hopefully that makes sense. So this type of mechanism can change the sustained membrane potential and impact over a longer period of time the excitability of the neuron, the likelihood that it's going to fire an action potential in response to any type of a stimulus that might be of the standard intensity stimulus. And in the case of decreasing sodium and producing a depression of the membrane, you push it farther away from threshold so that with the stimulus you're very unlikely to reach threshold and fire an action potential. But also remember, we looked at this in the same context as far as stimulus intensity in the previous presentation, and that when you got to a certain level, not only fired just one action potential, but you got multiple action potentials. So this is where you can turn a silent cell into a cell that might become spontaneously bursting because the membrane got so close to threshold that there's other ion channels in the membrane, like typically they're calcium ion channels, that now are going to just cause this spontaneous firing of action potentials, and you get a spontaneously active neuron that will be bursting. This is common of the dopamine neurons in the reward pathway. as well as in the motor pathway. As you might know, dopamine neurons degenerate in the substantia nigra of the midbrain in Parkinson's disease. So those neurons, when we decide to fire them, to initiate a movement, we do that to them so that they become spontaneously bursting, and they keep driving and motivating our behavior. So that's the way you want to think about that. This second messenger pathway system also modifies existing proteins or regulates the synthesis of new proteins to produce a coordinated intracellular response. One aspect of this, other than thinking that it might predict some long-range type of an effect that you need to boost up or bolster the cell mechanisms to be prepared for, is also to think of this in the way that this signal tells the nucleus that we just expended the activities of some proteins, and you need to express the genes to replace some of those. Another way that this is affected inappropriately is that if this is a substance of abuse acting through this means, then it produces not a coordinated intracellular response, but an inappropriate intracellular response, and sets the cell up to depend on, to crave the drug to be satisfied at its synapse, if you want to think of it that way. And that's anthropomorphizing a neuronal membrane a little bit, but it helps you to understand that this signaling to the nucleus and other proteins in the cytoplasm becomes very important in the normal function of cell recovery to a challenge or a demand placed upon it, a predictive change in a direction to sustain an enduring demand, right? As we get into shape, our cells respond better, right? Our neurons can help us run 5 miles better after 5 months of running 5 miles a day than that first day, right? And as well, they can be involved in replacing what is expended, and then finally in an uncoordinated, inappropriate response to some type of external drug or toxin that creates a pathological state, if you will. So now we can focus more so just on that inactivation. So we talked about the enzymes present either in the postsynaptic space, postsynaptic element on the postsynaptic membrane itself. But even though it's not mentioned in the text, I do know from the scientific literature that there are some enzymes out in the interstitial fluid that are there to degrade them if they escape into the interstitial fluid. So when they get into the interstitial fluid, they can be washed away in the blood. And they're not. Oh, they got the glial cell over here. Thank you. There we go. Can be taken up by glial cells. They can be degraded by postsynaptic enzymes. Number one here, we should go back. They can be taken up by presynaptic proteins, transport proteins, and processed, as I said, either degraded. They now become available for nearby enzymes to degrade, or VMAT, or vesicular transport proteins to recharge vesicles. So obviously after this vesicle has released its content, when it does come get back into the axon terminal, it'll be empty, so it's got to be recharged, whether it is recreated through endocytosis over here, or it's the kiss and release, where it kisses, creates the pore and empties out and then comes back in. Either way, it needs to be recharged to the homeostatic level of synaptic content. So that competition, if you will, it's concentration dependent here, between the vesicular transport to recharge and the degradative enzyme, contributes to the basal state for the rate at which you are providing transmitter for release. So the removal is a very important fast mechanism. The degradation is a very important fast mechanism. And then the diffusion out is a bit of a slower process, But it is very important. As I said, it can find receptors that are distributed along the membrane of the axons or the soma and the dendrites. And when those receptors pick up the signal there, then they know that there's a volume of release. It's called the volume transmission, as I told you, that is overpowering the synapse, if you will, and getting enough out into the interstitial space such that it can make its way up and bind there.